Hey, welcome back. It's Hampton here to talk to you this week about DNA replication in prokaryotes. Well, first things first, I need to find a prokaryote, which just so happened to be anywhere and everywhere around us. I guess I'm pretty lucky because miraculously an egg is on my whiteboard, which is full of E. coli, a prokaryotic species. So this might be an excellent time to start talking about DNA replication. In order for replication to occur, the following proteins and enzymes are necessary. Without any of these enzymes and proteins, this would not be possible. It turns out that there are specific nucleotide sequences called origins of replication where replication begins. Prokaryotes have a single origin of replication on its one chromosome. The origin of replication is recognized by certain proteins that bind to the site. An enzyme called helicase unwinds the DNA by breaking the hydrogen bonds between the nitrogenous bases. ATP hydrolysis is then required for this process. As DNA opens up, Y-shaped structures called replication forks are formed. The two replication forks are formed at the origin of replication. These get extended bidirectionally as replication proceeds. Single-stranded binding proteins coat the single strands of DNA near the replication fork to prevent the single-stranded DNA from winding back into a double helix. DNA polymerase is able to add nucleotides only in the 5-3 direction. A new DNA strand can only be extended in this direction. It requires a free 3-OH group to which it can add nucleotides by forming a phosphatidized bond between the 3-OH end and the 5-phosphate on the next nucleotide. Basically, that means it cannot add nucleotides if a free 3-OH group is not available. Then how does it add the first nucleotide? The problem is solved by primers that provide a free 3-OH group end. Another enzyme, RNA primase, synthesizes the RNA primer that is about 5 to 10 nucleotides long and a complementary to the DNA. Because the sequence primes the DNA synthesis, it is approximately called a primer. DNA polymerase can now extend this RNA primer, adding nucleotides one by one that are complementary to the template strand. This continually synthesized strand is the leading strand. The other strand, complementary to the 5-3 to parental DNA, is extended away from the replication fork in small fragments known as Okazaki fragments, each requiring a primer to start synthesis. This is the lagging strand. A protein called the sliding clamp holds the DNA polymerase in place as it continues to add nucleotides. The sliding clamp is a ring-shaped protein that binds to the DNA and holds the polymerase in place. Topoisomerase prevents the overwinding of DNA double helix ahead of replication fork as the DNA is opening up. It does so by causing temporary nicks in the DNA helix and resealing it. As synthesis proceeds, the RNA primers are replaced by DNA. The primers are removed by X-nuclease activity of DNA polymerase 1, and the gaps are filled in by deoxyribose nucleotides. The nicks that remain between the newly synthesized DNA and the previously synthesized DNA are sealed by DNA ligase that catalyzes the formation of phospholizer linkage between the 3-OH end and one nucleotide and the 5-phosphate end of the other fragment. Termination of DNA replication occurs when the two forks meet and fuse, and creating two separate double-stranded DNA molecules. The end. Now I'm going to go eat all this candy now. References.